DNA vaccine. How does this actually work? What are the advantages? So the example here is shown for the hemagglutinin from flu. You can take the gene that encodes that protein, put it into a plasmid, and then instead of injecting the protein into someone, you actually inject the plasmid. The DNA gets picked up by our cells, and then our own cells make the hemagglutinin protein, which they will also secrete. So the advantage here is that if the protein's made within our cells, that means it can get presented on MHC class 1, and that means you can also get a cytotoxic T cell response generated to that protein, as well as an antibody response, which you could get from a subunit vaccine. So the big advantage of DNA vaccines is to, one, get a cytotoxic T cell response, but also when you're injecting the plasmid and our cells are making it, at least for a while, there's antigen around for a longer period of time than just injecting the protein, which would actually disappear from our bodies very quickly. Some will get picked up by those innate immune cells in your skin, but then it might get into the bloodstream and your body will just chew it up and dispose of it. Here, at least, for a short amount of time, this plasmid is not designed to integrate into your DNA, so when the cell divides that it gets into, it will get lost. So it's not a permanent thing, which is safer, but it's longer than just giving protein, and that gives your immune system a chance to see it for a longer period of time and hopefully develop a stronger and memory response to it. And this just shows what happened in an experimental system in mice, that if they were immunized with a vaccine, they were protected. Of course, if they're not immunized, they're not protected. In a similar way, you could go even farther with the DNA and actually generate a viral vector vaccine. So if you remember back to the beginning of the lecture, I talked about cowpox being used as a vaccine. So that's vaccinia virus. If you take vaccinia virus, which we know can infect people and generate strong memory responses to that particular virus, what if we put in a foreign gene from a different pathogen? Then your body can also develop an immune response to that foreign protein, which comes from a different pathogen. So you would actually get protection from smallpox, because we know vaccinia can do that, but then also to whatever other infection this foreign gene came from. Now, this is a lot like an attenuated vaccine, because the vaccinia virus, although it's not truly attenuated, it's attenuated when it's inside people because it doesn't make us very sick. It does infect cows very well still. And it's also got a piece from another pathogen. So you can get cytotoxic T cell responses, but you're likely to get a stronger response overall because, again, this is a real infection, and your immune system is designed to respond to that. With your innate cells at a barrier site where you get exposed to that infection, and then activation in lymph nodes, and generation of both T and B cell responses, and the generation of a memory response. And remember, we already know that we generate strong memory responses to this vaccinia virus. So then we should generate a strong memory response to any additional antigen that happens to be included with that vaccinia virus. And that's the advantage over a DNA vaccine, where there isn't really an infection and there isn't really anything that causes inflammation that would give you a stronger immune response like you actually get with an infection. Another new way of developing vaccines, again, is to try to trick the immune system into thinking that it sees an actual pathogen. And that's shown here with this virus-like particle. If you are infected with a virus, remember your innate immune cells, using the receptors on their surface, will bind to the PAMPs that are on the surface of an infectious organism and internalize that and eat it. those innate immune cells, like a dendritic cell or macrophage, don't know that it doesn't have DNA inside and can't replicate. On the outside, it looks just like a virus. So they're going to be strongly activated in response to this particle. But this particle is actually safe because there's no DNA inside. You do not actually get infected, but you are likely to get a good, strong innate immune response. And remember, it's your innate immune response that turns on your adaptive immune response to get B and T cell memory responses. Now again here, because these proteins aren't made within the innate immune cells and there's nothing really infecting our cells here, you're not going to get a very strong cytotoxic T cell response, but you do have the potential here, and a better potential than just giving protein alone, to actually get cross-presentation, which you have learned about in a previous lecture. Another new advance in vaccine design and development is to actually focus just on the epitopes that stimulate the immune response. And this is just a picture of that shown here. Remember, in a whole protein, only a very small piece might get presented on either class 1 or class 2. Most of the protein doesn't have the right amino acid sequence in order to bind to an MHC molecule. So you don't even need to give an entire protein. You only need to give those small epitopes that are actually what MHC and T cells then interact with. And you can choose either peptides that would bind to MHC class 1 or class 2 or both. And along the same lines, remember that antibodies generated to a particular protein antigen, you will have many different antibodies generated to different epitopes. Some of these epitopes might be linear in the protein, but some of them might be conformational, such as shown here, where this epitope is created by the protein folding and two pieces of the protein coming together that are not sequential in the amino acid sequence. Now, this is much harder to develop an epitope vaccine for because you can't bring those two pieces easily together. And so if you just want to use epitopes to generate antibodies, you need to find linear epitopes. But this also brings up an important point that when you do things like recombinant proteins, you do need to try to make sure that the protein is in its right conformation that you're using for your vaccination. When you make a recombinant protein, it doesn't always fold up the same way it would in its natural environment within the pathogen. So if this antibody is important for the immune response and the protective response to that pathogen, you need to make sure that this conformation of this antigen is what the immune system actually sees. Now, of course, if you have an attenuated vaccine, you know it's sort of going to be in the right conformation. But whether it's a killed vaccine or a subunit vaccine or even a conjugate vaccine, you don't necessarily know that. So that's why sometimes vaccines don't work. In particular with subunit vaccines, it's because we don't have the right conformation of the antigen. And that matters, again, in particular for antibody responses. For T cells, it's always going to be a peptide presented on MHC, so conformation of the entire protein doesn't matter. But it does matter for antibody responses. Okay, so we've gone through a lot of different formulations of vaccines. How do you figure out how well they work and what things are important to consider? Do you generate the right sort of immunity? What I mean by that is, do you generate antibody or cell-mediated cytotoxic T cells? How about in the correct compartment? Again, if you've got a pathogen that is a fecal-oral route, you need actually a mucosal immune response. For other pathogens, if it's the flu or other respiratory infections, you want to make sure you have a good immune response that's going to actually work in your lungs. Now, for the vaccines that we have that work, they seem to generate strong systemic immune responses that do end up working in the appropriate place for where the pathogen actually exists in the body. But that is also a limitation in why we don't have vaccines for as many diseases as we would like. And then, of course, are you choosing the best antigens? Again, if you do an attenuated vaccine or a whole-killed vaccine, you've kind of got all of the antigens there. But a lot of our new vaccines are moving to small pieces of a pathogen, and then you have to do more work to make sure that immune response to that particular piece of the pathogen will actually give you a protective immune response to that whole pathogen. Then there's the issue of, do you generate a strong enough immune response? Again, an attenuated vaccine, it's not a problem. Viral vector vaccine, all of those are infections, and our immune system is designed to respond strongly to infections and create strong immune responses and also memory response. But when we're trying to do things with injecting proteins or just DNA or proteins linked to carbohydrates, we're sort of trying to trick the immune system, and sometimes we just don't get a strong enough response. And then again, you have practical concerns, which I'm not going to spend too much time discussing. So, you have to pick the best antigen. You have to have a way of trying to ensure that you're getting the right type of immune response, whether it's antibody or cell-mediated or compartment. You have choices for the route of delivery. Most often, they're intramuscular, but again, they can be intranasal, they can be oral, subcutaneous, so really in your skin, but not into your muscle. How much to give? How long do you actually get antigen presentation? That will relate to adjuvants that we'll talk about. How many vaccines you get, how many booster shots you get, and then, of course, what's the age and immune status of the recipient. For instance, we know that with the flu vaccine, the elderly do not generate the same type of response as younger people do to the same vaccine. So one thing that's important for vaccines is boosting. And if you've gotten some vaccines, something like measles you get once and you don't need a booster. Again, that's an attenuated live viral vaccine. But if you got the hepatitis B vaccine, you got multiple shots. That's a subunit vaccine, and you need to show it to the immune system multiple times, and eventually you'll get a strong response. And that's just what these graphs are showing you here. The other thing that happens with boosting is that you will switch from an IgM to an IgG antibody response. We learned about that in your B-cell lecture. And also your antibody affinity will increase with somatic hypermutation. And again, which types of vaccines require boosting? Anything that's not a live infection is going to require some form of boosting, because only the live infection is really what mimics what happens during a real life infection. And all our other vaccines don't. So we have to keep reminding the immune system that they really do want to make an immune response to our particular vaccine. So that brings me to adjuvants. What are they? They're substances that enhance immune responses to antigens. We know about a lot. Our limitation is not finding adjuvants, it's finding safe adjuvants. If we overstimulate the immune system, we could actually cause problems in somebody, cause somebody to go into shock, have too high of a fever, too many cytokines. So that's the limitation with adjuvants. Their job, three main jobs. One is what we call create an antigen depot. So I told you that when we inject a protein, one of the problems is it gets picked up by a few innate immune cells, but then it disappears and our body degrades it and gets rid of it. And then it doesn't give enough time for the immune system to really see it and make a response. So can we slow down the release of the protein to overcome that initial problem? Can you use adjuvants to target your antigens more specifically to those innate immune cells that are found at barrier sites? And then can you modulate or enhance the type of immunity? Whether you want different types of T cell help, for instance, which is important for the type of antibody you might generate, and that matters for the type of infection, whether you want more of a Th1 or a Th2 type antibody response. So, why do we need adjuvants and for what types of vaccines? Again, anything that's not a live vaccine, so not attenuated and not a viral vector vaccine, needs an adjuvant. Benefits, again, you can use less of your antigen. So you can save money and time in preparation by using less. You may need fewer booster shots. You can enhance the way the vaccine works in people whose immune systems are weaker, either because they're very young or very old or have some sort of immune deficiency. You can increase the total antibody titer you get, which is likely to give you a stronger protective response. You're more likely to get a longer lasting immunity. You can sometimes provide a broader protection by generating, again, a stronger immune response. You can use certain types of adjuvants that might facilitate more specifically mucosal immunity. And you can overcome something called antigen competition. Sometimes we try to combine multiple antigens into one vaccine, like in that viral particle I showed you that had multiple different antigens on the surface. Sometimes what happens, though, is that the immune system focuses on just one when we really want an immune response to all those proteins. And adjuvants can help to facilitate that. So, adjuvants can target different PAMPs. Remember those pattern recognition receptors on innate immune cells. But first they can serve as that antigen depot. Slow down the release. Don't just dump the protein into the body. It will disperse and disappear, and most of it won't get picked up by the immune cells. So slow down the release. Have adjuvants that bring the protein very specifically to those PAMP receptors, such as TLRs on the surface, or also TLRs that are intracellular. And this can happen through inflammasome activation. And again, you want that because you want to generate a stronger immune response. And then your adjuvants can also help you with increasing and improving antigen presentation, and that's what's ultimately going to help you get a stronger adaptive immune response and a stronger helper T cell adaptive immune response. Okay, so the most common adjuvant we have worldwide is something called alum. But until recently, just in 2011, we didn't actually know how it worked. And what I showed you here was more specific ways of targeting immune receptors on the surface of cells and inside cells. But actually, alum does something different. Alum does not bind to receptors on innate immune cells. The alum actually binds to the cell membrane itself. So it binds to the lipid. Grease, of course, is fat, and lipids are fat. So the alum sticks to the cell surface, the innate cells. And that's a different delivery method of trying to get the cells to take up antigen. And although we may not have predicted that would work, it turns out to stimulate those cells very well and allows you to get antigen inside of cells and presented. And this actually turns out to be a better mimic of infection and having pathogens inside of cells than even just targeting receptors on the surface. So alum is not our strongest adjuvant. It is by far our safest adjuvant, and it works in a way that we did not actually expect. Okay, So I want to go back and talk about the steps to developing a vaccine. First, you have to define your disease and actually define your pathogen. Now, that sounds simple, but for some pathogens, we don't actually have a good handle on that and know exactly what the disease is or exactly what the pathogen is. For instance, we may not have been able to isolate the pathogen. If we can't grow the pathogen in vitro or isolate it, we know it exists, but it's going to be very difficult to develop a vaccine if we can't isolate and grow a pathogen in vitro. We need to have some sort of observed immunity. Can you develop immunity to whatever that pathogen is? Again, with a vaccine, we're trying to mimic what happens naturally. So we need to try to understand how the immune system normally responds and what type of memory response is generated. So our most successful vaccines so far have been against pathogens for which we very clearly develop a protective memory response. If you get smallpox once and survive, you do not get it again. If you get chickenpox once, you do not get it again. Those pathogens provide us with a very clear pathway of what type of immune response can be generated and how good it is at providing protection. But not all pathogens are like that. Malaria would be a very good example where an individual can repeatedly get infected by the parasite. And they can develop something called naturally acquired immunity, which gives you protection from disease but not infection. So it's one of the reasons that it's been so hard to develop a malaria vaccine. With our observed immunity and correlates of immunity, we don't have a good immune response to try to mimic with a vaccine. We're trying to beat nature when we try to create a malaria vaccine. Now, again, with correlates of immunity and critical antigens, we know that our bodies will respond to many different antigens on a pathogen. It does not mean, though, that all of those immune responses actually play any role in protection. So often it's just one or two antigens and sometimes one or two epitopes against which the protective immune responses are generated. So you don't just want to pick the most abundant protein on a pathogen as a subunit vaccine. You do want to have some kind of understanding about whether immune responses to that antigen do actually give you any protection. And again, that's why we don't have as many subunit vaccines as we would like. We can generate vaccines that stimulate immune responses. It doesn't mean those immune responses can always protect you from infection. One thing that's really important for vaccine development is having some sort of animal model. If you can't test your vaccine in animals, you don't have a good way of knowing if it can actually afford protection. Again, you can determine if an immune response is generated, but you cannot correlate an immune response with protection. And an animal model can show you that an immune response is actually protective or not. You need some then sort of proof going beyond an animal model in people or in animals if your vaccine was designed for animals, for cats and dogs, for instance, or maybe for cattle on a farm or pigs on a farm. Of course, you need to figure out if it's safe. You then need to do clinical trials to figure out if it works, and then ultimately you need to try to get approval for the antigen. This process can take decades if it's a disease for which we do not have a vaccine. So currently there is a relatively new malaria vaccine that doesn't work particularly well, but it's better than anything we had in the past. The antigens used in that vaccine and the idea to make that vaccine started way back in the 1970s and 80s. And it's only in the last few years that that vaccine has even made it to market. So we're talking about 40 years. Now, when you have a vaccine like the flu vaccine, the system is set up and we can create a new pool of killed flu virus organisms every year using the same system. We just grow up different versions of the flu depending on which flu isolates we think will be most common throughout the world to use for the vaccine. So, today, we've gone through a lot of different vaccine designs. But remember, overall, the job of a vaccine is to try to mimic protective memory responses that would occur naturally if you got that infection. So we went through an attenuated vaccine, which is alive and actually infects you, but is weak, so does not make you sick. And that vaccine almost always works very well with one vaccine. You do not need boosters, you do not need adjuvant, and it's very long-lasting because that vaccine most closely mimics what happens during a natural course of infection, primary stimulation of an immune response, generation of memory, all the things you've learned about in your previous lecture, and then protection if you were to ever encounter that pathogen. Whole-killed vaccines we talked about, kind of quick and easy and also dirty, but they can be made, and if they can generate good antibody responses, if antibodies are protective, then those vaccines can work well. Subunit vaccines, that's taking one protein from a pathogen and using that to generate a vaccine. You almost always need an adjuvant with that, because if you just inject a protein in somebody, it is foreign, but it doesn't hang around for very long. You need the antigen to slow down the release of that protein and also to really cause a stronger inflammatory response to get those innate immune cells at the barrier sites that are there to see foreign antigens to really be more interested in that one protein. We talked about a conjugate vaccine. That's bringing together protein and non-protein components. In that case, a polysaccharide and a protein. And that's so you can get T-cell help for a non-protein antigen. In nature, that doesn't occur within one antigen. It can occur, of course, within a pathogen, because a pathogen will include protein and non-protein portions. So we're trying to mimic the pathogen by bringing together a piece of a protein and a polysaccharide in one molecule instead of the whole pathogen. We also talked about some of those new vaccines, like DNA vaccines and viral vector vaccines, and polyvalent viral particles. All of those are new ideas, basically trying to do a better job of mimicking protective memory responses and taking advantage of all the knowledge we have from attenuated and whole-killed and subunit vaccines to try to create a smarter vaccine that really does a better job of tricking the immune system into thinking it's seeing a real infection when actually it's not. So, again, all the vaccines I just mentioned, they stimulate the immune system in a different way. So you have to think about the different vaccines, how they're actually stimulating the immune system, what's working well, what's not, and then, of course, can you get cytotoxic T-cell responses as well as antibody? Because again, just about every vaccine we have will give you some kind of antibody response, but only a subset of them can also give you a cytotoxic T-cell memory response. We spent some time talking about adjuvants. They're necessary for many different types of vaccines. Anything that's not an actual infection will need an adjuvant. An infection, because it's growing and dividing in your body and is a whole organism, is its own adjuvant. It has those receptors that stimulate PAMP. You don't need to slow down antigen delivery because infecting multiple cells in multiple rounds means you get that prolonged antigen exposure. So you only need an adjuvant when you don't have a true infection, which would be an attenuated vaccine or a viral vector vaccine. I hope you enjoyed learning about vaccines today, and basically vaccines incorporate all of the basics you've learned about how the innate immune system works, how it activates the adaptive immune system, and how B and T cell responses are generated in a primary exposure, and then also to generate memory. Thank you.